Yes, this session is on early career investigators and trainees. And Dr. David Stevenson and I will be the moderators. The first speaker is Dr. Kevin Contrepois, who is the scientific director of the Metabolic Health Center in the Department of Genetics. His paper is on global urine metabolic profiling for prediction of gestational age. Kevin?
So yeah, thank you so much to the organizers for giving me the opportunity today to present some of our work really around gestational age dating using urine metabolites in term and preterm pregnancies. So first of all, why is it so important to accurately estimate gestational age? It's because it's actually a critical piece of information used in the clinic to provide optimal care for expectant mothers and also inform on clinical decisions. And this is especially true for intrauterine growth restriction and also preterm birth. So the gold standard to measure gestational age is actually by using fetal ultrasound, ideally performed before 13 weeks of gestation. However, this type of clinical test is often not feasible in resource-limited settings due to later presentation to care or lack of equipment or trained sonographers. So there has been alternatives to estimate gestational age, including first day of last menstrual period or other measurements, but all of these methods are actually imprecise and even biased in certain cases. So recently, in the past 3 to 4 years, there has been many omics studies performed in blood from pregnant women, and we actually could detect very finely tuned chronological changes with pregnancies. And this laid the idea of being able to predict gestational age using molecular markers. So this was done at the level of cell-free maternal RNA, also immune cells, plasma proteins, and lastly, metabolites. That was actually published by our group earlier this year in Cell. So while most of these omics layers actually demonstrate a very strong predictive value, metabolomics was actually one of the most performant approach to be able to estimate gestational age. And we actually showed that in a publication last year by comparing the different predictive ability of the omics layers. So now we know that we can predict gestational age from blood, but what about urine? And urine is an obvious advantage in the clinic because it's non-invasive and also it doesn't require any processing. So this is why this study was designed. And this was done in collaboration with 2 international biorepositories called Amani and GAPS, where urine samples were collected from 4 different countries among 5 different sites. So in Bangladesh, Pakistan, Tanzania, and Zambia. So we received 99 urine samples that were collected early in pregnancies between weeks 8 and 19. And upon reception, we actually ran our untargeted metabolomics platform that uses 2 different liquid chromatography as well as high-resolution mass spectrometry. So these methods here have really been designed to try to have a broader coverage as possible, measuring as many molecules as we can from these urine samples. And after data processing and curation, we ended up with over 6,000 individual metabolic features that were used for downstream analysis. Looking globally at the data, we actually could see on the left plot here, on the principal component analysis, we could see that the samples could be ordered already by gestational age. And these were along principal component 1 and principal component 4. And this really demonstrates already that there is a lot of metabolite signal here in urine that can track very well gestational age. When we looked a little closer into the biological processes that were enriched among the molecules that can track pregnancy, you can see on the right side, steroid hormone biosynthesis is by far the pathway the most enriched. And this is totally expected, and it's a nice validation here for our analysis. So next, we looked at the individual molecules that can associate with gestational age. And you can see on these volcano plots that most molecules are actually on the right side, meaning that they positively associate with gestational age. And this is a nice validation from our analysis because we can recapitulate some results we have demonstrated from plasma, including some estrogen derivatives, as well as progesterone derivatives, corticosteroids, and androgens. So here we find actually the same molecules that we've described in our Cell paper earlier this year, and we also find them in urine. So this is validating our approach. But what's interesting here in urine is that it's a very complex biological matrix with many derivatives of metabolites that we might not see in blood, but that we can detect very easily in urine. And as you can see at the top of this plot, we have two molecules that are actually unidentified. The first one contains 19 carbons, 20 hydrogen, 1 sulfur, and 8 oxygens. So this molecule is a steroid-like molecule that is modified with a sulfate group, but it's unidentified. We don't know what it is. And the second top molecule here is also an unidentified molecule. So here it's very interesting that we can recapitulate the results from plasma with some non-molecules, such as estriol glucuronide, but also there is some signal from unknown hormones here that shows even more predictive. So in order to move closer to a clinical setting, we actually generated a predictive model that contains as few metabolites as possible, and we use random forest to do that. And we ended up with a model using the top 3 metabolites that yielded an excellent predictive ability, as you can see on the scatter plot here, that shows the predicted gestational age versus the observed gestational age. So the raw value is close to 0.9, and the root mean square error is 1.58 weeks, which is really an excellent performance here for our models. Importantly, we actually validated this model on a totally independent cohort that was enrolled at Stanford. So this cohort here obviously have a different ethnic background, but even though the ethnicity was different, we can reproduce very nicely here our predictive model. So what I didn't mention earlier is this study actually contains samples collected from pregnancies that went to term, and half came from preterm deliveries. So we asked the question, can we estimate gestational age as well in both of these cohorts? The answer is no. You can see very clearly on this scatter plot on the left side that the term delivery, the model for term deliveries is actually much more performant than the preterm delivery model. So we ask ourselves, what can be the reason for that? And one explanation we have is that the top metabolites that track pregnancy progression actually have a higher interindividual variability in preterm pregnancies. And this was especially the case between weeks 14 and 17. As you can see here on this box plot showing the coefficient of variation of the top metabolites, we have a higher variation for the preterm deliveries. So this can explain why the model doesn't perform as well in preterm deliveries. And importantly, we also compared the relative abundance between term and preterm, and abundance of these top features are actually very similar. So it's not so much the abundance, it's more the variability, interindividual variability. So to conclude, we demonstrate here the feasibility of implementing urine metabolomics analysis in multi-site studies. We also report a predictive model for gestational age that has high accuracy and uses only 3 metabolites. And this model was validated using an independent cohort, and the predictions were not as robust in pregnancies that ended prematurely. And to conclude, our approach really showed great promise and could benefit pregnant women worldwide because collection of urine is non-invasive and it doesn't require any processing in comparison to blood that is harder to get and require processing. So finally, I would like to acknowledge everybody involved in this study, including the Snyder Lab, as well as the Prematurity Research Center, and all of the funding agencies that enable to make this work possible. So thank you very much for your attention.
Kevin, wonderful presentation. There's one question that came up, and how stable are the 3 key metabolic metabolites in the field? So
stable in what sense? In urine for degradation?
How stable are the key 3 metabolites in the field? It says high heat. I'm not 100% certain what they're...
I mean, Yeah, so here when we measure metabolites, some actually are susceptible to degradation. This is why it's so important to actually have some SOPs to collect urine and freeze it keep it at 4 degrees and freeze it as soon as possible. So here, among all of these sites, urine samples were actually collected in the clinic and frozen very quickly within 2-hour window. Otherwise, it was kept at 4 degrees. So this is ideal setting to measure molecules. And because these molecules are steroid hormones, these are typically considered relatively stable.
I have a question. What happens with the dietary profile of the mother? If mother has malnutrition, is that going to change your profile?
That's right. It's definitely a possibility. And this is a challenge of working with such metrics like urine. Here we didn't take it into account, but you can see that these molecules track very well. So I think it will be valid in any case scenario.
Great. Thank you for your presentation.
Thank you so much.
Our next presenter is Dr. Inda Stelzer, who is a postdoctoral research fellow in anesthesia, perioperative pain medicine at Stanford. The title of her presentation is the Integrated Trajectories of Maternal Metabolome, Proteome, Immunome Predicts the Onset of Labor. Dr. Stelzer.
Good afternoon, everybody. Thank you very much for the introduction. Before I start, I would like to thank the committee for giving me the opportunity to present our multidisciplinary study at this symposium. Actually, Kevin's presentation is a very nice segue into our study, in which we also measured the maternal metabolome, although this time in the plasma. In the next few minutes, I will talk about a new approach to predict the due date. We know from decades of immunological work that the maintenance of pregnancy relies on a tightly regulated immune balance. However, most of the knowledge that we have is based on local adaptations that occur at the fetal-maternal interface. The research in our lab focuses on the systemic changes that occur over the course of pregnancy in the peripheral blood, which is a tissue that's accessible in the clinical routine. And we do this to understand profiles of the progression of pregnancy. We are measuring these immune system dynamics throughout pregnancy using mass cytometry. And this high-dimensional platform allows us to capture all major innate and adaptive immune cell population frequencies and their intracellular signaling capacities, endogenous as well as after stimulation. The correlation network of these features can then be leveraged to predict an outcome of interest. And in collaboration with Nima Aga-Epur's group, we are using multivariate algorithms to train prediction models of these outcomes. This approach resulted in the description of the immune clock of a healthy pregnancy, where a regression model predicted the gestational age from maternal blood with high accuracy and revealed immune dynamics across pregnancy. In the study I'm presenting today, we aim to use our ability to predict gestational age and expand it towards a clinical question. Can we predict the time of delivery? And as we've just already heard from Kevin, in the current clinical practice, the delivery date and the gestational age is calculated based on an estimate that is derived from the time of the last menstrual period and then confirmed by ultrasound during pregnancy. And this calculation assumes a fixed progression of 40 weeks of gestation. However, most pregnancies actually deviate from this norm, and especially in preterm or postterm pregnancies, it's clinically of very high importance to be able to say when parturition is going to happen. Currently, our tools for predicting the onset of labor perform rather poorly, though, so we really need novel estimation approaches, including predictive biomarkers, to better anticipate parturition. To this purpose, in our study, we in contrast, used the time to delivery to capture the progression of individual pregnancies. And this was independent of the duration of pregnancy itself. So predicting the time to onset of labor from blood can support us in our search for universal pre-labor mechanisms and potential predictors of delivery that are shared between term, preterm, and postterm pregnancies. Since we know that the immune system does not work in isolation, we expanded our measurements to also include interactions with the proteome and the metabolome, for example, all the pregnancy hormones. So in this study, 53 pregnant women who received routine care at the Stanford hospital were recruited, and all of them went into labor spontaneously. We also had a test set of 10 additional pregnancies to independently validate our prediction model. For each study participant, we collected serial blood samples during the last 100 days before labor. And we defined a continuous variable, the days to onset of labor, which describes the difference between the day of sampling and the day of onset of labor, which you see here in this red line. We quantified the plasma concentration of metabolites and proteins using a high throughput untargeted mass spectrometry assay and an Aptamer-based proteomic platform. With mass cytometry, we extracted single-cell immune features from each sample. And this combined analysis gave us more than 7,000 features per sample, which were then integrated into a multi-omic model to predict the day to onset of labor. And this is a huge team effort. So Kevin and team generated the metabolome data, and together with Sharon, Sajjad, Kaz, and Julian, we are currently working on the revisions of this paper. So to investigate whether the combined data sets would model the time to onset of labor, linear regression models were built individually and then integrated via stack generalization. And this final model predicted the day to onset of delivery with high accuracy in an RMSE of 17.7 days, so the true versus the predicted day to onset of labor. And the generalizability of this model was established using a two-layer cross-validation strategy within this training set. Our model was additionally validated in this test set of 10 additional new patients. So the assessment of maternal circulating factors in the peripheral blood actually provides an accurate prediction for the timing of labor onset. And this is independent of an estimate of gestational age. To do the biological interpretation of the results, we focused on the model features that contributed most to the prediction of the DOL. And you can see these in the scatter plot here. These 45 features were distributed across the 3 omic data sets and included, for example, 17-hydroxyprogesterone, cortisol, and progesterone derivatives, which was a nice validation and a very expected outcome of our study to see an increase for example, cortisol right before labor. We also saw IL-1, IL-4 and proteins involved in angiogenesis and inflammation and coagulation, and immune responses in innate and adaptive population, which were dampened with approaching labor. For example, the STAT1 signaling response in NK cells upon interferon alpha stimulation. We then also performed additional analyses to better understand how these features would behave over time. And using a regression, another regression analysis, we identified a switch point of feature behavior at 23 days before labor onset. So our analyses indicate that the maternal metabolome, proteome, and immunome undergo a marked transition from progressing pregnancy into a phase of pre-labor biology, and this is linked to the timing of labor onset. So this pre-labor phase, which begins around 3 to 4 weeks before parturition, is characterized by stagnating innate immune responses, or in other words, a peripheral immune response regulation, and surging levels of IL-1, IL-4, progesterone, and cortisol. So in summary, our findings laid the groundwork for developing blood-based methods for predicting the time to delivery. And these are anchored in pre-labor mechanisms that are shared between preterm, term, and post-term pregnancies. With this, I would like to thank all study participants and my mentor, Brice, and the whole lab, as well as the ARA-EPO lab and Nima and Sajjad. I would like to thank Martin Angst and all researchers associated with the Stanford Prematurity Center led by David Stevenson. And also thank Kevin Contrepois and his team in the Michael Snyder lab. Thank you for your attention.
Thank you, Dr. Stelzer. Wonderful presentation. I'm checking to see questions. Can I ask one? As you proceed forward, how would you see this working in terms of cost and the difference that this would make clinically in the management of pregnant women?
Sure. So this study was really designed to describe the pre-labor biology that we so far really don't understand very well. So a next step would be to really distill from those 45 45 features that now inform the prediction model, really distill those that are most predictive, and then validate them in a set that includes more pregnancies and pregnancies of varying length as well.
Great. Well, I want to thank you for your presentation. Great work. Thank you. Our next speaker is Dr. Victoria Ward, who's a clinical assistant professor at Pediatrics, Division of Hospital Medicine. And she, the title of her talk is The Evaluation of Large-Scale Maternal and Child Health Programs in Bihar, India, through an Equity Lens. Dr. Ward.
Hi, I'm going to talk about an equity analysis of a large-scale maternal and child health program in Bihar, India. This work was supported by the Gates Foundation, but otherwise they have no disclosures. Equity has become an important focus over the last year, but it's been key to the global health agenda for a long time. SDG 10 calls for a reduction of inequality within and among countries, and the countdown to 2030 aims to improve women and child health by focusing on coverage and inequality of interventions. Our equity analysis focuses on Ananya, which was a Gates Foundation program implemented in Bihar, India. Bihar is a state of 100 million people, and it's in the northeast of the country. It also has some of the highest morbidity and mortality for women and children worldwide. Against this backdrop, the Gates Foundation implemented a $300 million program intended to provide key health interventions for women and children's health, and also to drive demand for those interventions through multimedia and frontline worker outreach. Their idea was to implement their programs in 8 districts of the country, or of the state, across 28 million people. Those that were found to be successful would then be scaled up across the other 30 districts for 75 million people. To do so, they implemented surveys, a baseline in 2012 and a midline in 2014. The endline was actually never done. Those surveys were large, about 20,000 people per survey. They also did a monitoring survey collection with Care India. Rounds of data were collected once or twice per year, and again, across thousands of women to really understand what was working and what needed to be changed. We studied these programs over the last 3 years to really understand what was most successful and for whom. Today's analysis that I'll focus on is an equity analysis, arguably most important because we studied who might have been left behind when these programs were implemented. Our aim is to examine whether program impact differed between the most and the least marginalized groups, and then to examine whether those health inequities between the two groups changed over time following implementation. We used an intersectionality approach, which is well documented in the literature. We combined socioeconomic indicators with CAS to create a 9-level comparison between the least and the most marginalized groups. This was across women who had infants less than 12 months, and it was a 3-stage sampling design to randomly select women, 13 per village. Ultimately, we were able to do a difference in difference analysis between the least and the most marginalized women, yielding a quasi-experimental program impact study. Briefly, this is our flow chart just to show that in the focal districts compared to the non-focal districts, in the baseline survey, there were 2,400 women in the focal districts and 7,800 in the non-focal districts, and that was similar for the midline survey. Demographically, the most marginalized women were, as you would expect, illiterate, less schooling, older, and smaller households. Turning to the data, we'll focus in on the indicator identification of a skilled birth attendant, a key birth preparedness practice that was highlighted in the program. For the least marginalized, 75% of women in the focal district, so where the program was implemented, compared to 68% of women where the program was not implemented in those control districts, were the numbers for the baseline. In the midline, that 75% number went up to 100% of women identifying a skilled birth attendant in the focal districts amongst the least marginalized women. The number went down in the comparison districts, yielding a difference of 33%. Now, for the most marginalized women at baseline, only 50% of the most marginalized women identified a skilled birth attendant. That went up to 65% and did not change in the comparison districts, but ultimately yielded a larger disparity between the least and the most marginalized women. We were able to compare this difference in difference analysis across 18 indicators and ultimately found that disparities increased favoring the least marginalized for indicators like skilled birth attendant, skin-to-skin care. We found that disparities decreased favoring the most marginalized for immunizations and intermediate breastfeeding. Turning to the Care Collected Household Survey. This was the survey that was collected in rounds, so it was collected throughout the period of program implementation from 2012 to 2017. Again, amongst women who had children that were 0 to 11 months of age, and sampling was done using a quasi-lot quality assurance sampling methodology. We were able to compare indicators across RMNCH and indicators for the least and the most marginalized women using odds ratios, and our models were adjusted for age and gender of the focal child using an FDR correction to account for multiple testing. Turning to the data, we can start to look at these indicators and what changed over time. So for the most marginalized, the red line at the bottom, really there was no change throughout program implementation for the most marginalized women. For the least marginalized, you see a slight increase over time. So ultimately, the disparity gap widened between those least and most marginalized women. For indicators like complementary feeding, least and most marginalized women generally perform this at the same rate at the beginning of the program. Throughout program implementation, that number increased for the least marginalized, and again, there was no change for the most marginalized. So again, a widening of disparity. When we look at indicators that have to do with frontline worker advice given, so were women receiving that advice from their frontline workers about that important practice? And really there was no change. The least and the most marginalized women reported that that advice had been given by frontline workers at the same rates. So while their information receipt wasn't different, their behaviors were. We looked at this across 23 indicators using odds ratios. And ultimately, when we look at it in aggregate, we found that disparity widened favoring the least marginalized for 7 of those indicators. Disparity decreased favoring the most marginalized for 4 of the indicators. And there was no disparity in the indicators related to frontline worker advice given. When we start to look at these indicators by category or by intervention platform, we start to see something interesting. On the left-hand column, you see that frontline worker given advice was not different between the two groups. On the right-hand column, however, you see quite a bit of disparity widening for all of those indicators that are associated with facility-based delivery or service outreach. In the center column, where it says maternal-driven behaviors, there's a mix. However, when you drill down into the indicators, you start to see that those indicators that are related to access to transport or varied sources of food, those are where you see the bigger disparities. And those indicators that are related to maternal choices, like immediate breast or like breastfeeding, those do not change. Ultimately, we find that disparities in health-related behaviors were present at baseline and persisted throughout this program implementation. Disparities increased in facility-based services, and they decreased in some maternal-driven indicators, which were not reliant on access. The study was certainly limited in that of all self-reported surveys and thus certainly prone to social desirability bias and response biases. The surveys were collected as monitoring data, and the evaluation survey by Mathematica did not have an endline at the very end of the program implementation. Furthermore, our analysis was limited to extremes of equity groups. We find, however, that this demonstrates how important it is to do equity analyses for any program implementation and evaluation. Assessment must be performed to understand what individual and system-level barriers there are to access and equity during program design. And these impacts should be discussed across program funding and government stakeholders. Thankfully, we had the opportunity to share these findings with our colleagues at the Government of Bihar and the Government of India, as well as our Gates Foundation program officers and our program implementation partners. Special thanks to the Ananya Study Group, Gary Darmstadt, our PI, and the rest of the team. The findings of all of these will be distributed in a 13-paper supplement in the Journal of Global Health coming soon.
Thank you, Dr. Ward. Let me ask a question. Once you've done this work and you see how it comes out that there is these disparities and very hard to change, what would you recommend? What would be the next step to take for people that want to do improve global health for these populations?
Yeah. Thanks, Fernando. That's a great question. I think interestingly, when we circle back to the initial program design, we find that the idea was to be agnostic across groups and to distribute these programs widely across huge populations. There was not a focus on who might be the most marginalized or who might be left behind during program implementation. And what we find is that if you don't specifically focus on those groups and try to get an understanding of who might not receive the program, we see this across our mHealth studies, we see this unless the group is specifically looked at to figure out that their access will be present for them, then you find that there is disparity. And so really what we've promoted is that it can't take a group agnostic approach. You really have to focus on who might be left behind.
Great. Thank you. Great presentation. I'm going to turn the session over to Dr. David Stevenson, who's in Stanford hallways there, and let him take over.
Thank you, Fernando. Let me offer you first my congratulations. I've known you for over 40 years and worked with you over that time, and it's a well-deserved honor. And congratulations also to the preceding speakers. I have the pleasure of introducing the last two speakers in our session. And our first speaker is Sharon Paige. She is an instructor in pediatric cardiology. She's been the recipient of MCHRI Instructor K Awards support. And the title of her presentation is Mitochondrial Dysfunction is Associated with Impaired Contractility in Hypoplastic Left Heart Syndrome. Dr. Paige.
Okay, excellent. Well, thank you for the introduction, and thank you to the organizers for inviting me to speak today. So I'm going to be sharing with you some of the work that our group has done investigating hypoplastic left heart syndrome. So to give you an overview of this disease, there is a severe underdevelopment of the left side of the heart. Specifically, there's severe stenosis or atresia of both the mitral and aortic valves. There is hypoplasia of the ascending aorta, and there's also profound underdevelopment of the left ventricle. And this ultimately results in the left side of the heart being unable to support the systemic circulation. And what happens is that these babies who are born with HLHS develop profound circulatory failure and ultimately death within the first couple of days of life if this is left untreated. And while there's been remarkable advances in the medical and surgical treatment of this disease, patients still face significant morbidity and mortality, often related to failure of the single right ventricle. And ultimately, cardiac transplant remains the only definitive cure for this disease. So therefore, a better understanding of what causes HLHS and its associated complications is needed to help identify strategies for preventing and treating this disease. So while the precise etiology of HLHS is not fully understood, we do have some important clues. In a subset of patients, a genetic predisposition can be clearly identified, such as Turner syndrome, and also non-syndromic mutations in genes like Notch1. In addition, even in the absence of an identifiable genetic mutation, among patients with HLHS, there's an increased risk of bicuspid aortic valve in first degree relatives. And this indicates that there is a significant heritable component of left-sided heart disease. We also commonly talk about the consequences of what we call the no flow, no grow theory during embryonic development. And this would lead us to believe that reduced blood flow through the left side of the heart likely secondary to valvular obstruction, is what causes HLHS. However, the specific contribution of the cardiomyocytes themselves to the pathogenesis of HLHS, as well as the subsequent failure of the single right ventricle, remains largely unknown. So to answer this question, I turn to induced pluripotent stem cells, or iPS cells, which represent an attractive model system for studying human disease. Any somatic cell type, usually skin fibroblasts or peripheral blood mononuclear cells, can be reprogrammed to a pluripotent embryonic stem cell-like state, and then they can be differentiated in vitro into any cell type, including cardiac cells, that can then be used for various assays. So for the experiments I will show you today, the iPS cells were derived from 3 children with HLHS who had early unexplained failure of the single right ventricle, and the control group includes 1 healthy parent from each patient, as well as 2 unrelated healthy controls. So this is to give you an example of the cells that we routinely generate in the lab, and you can see this almost ocean wave-like pattern as the cells have differentiated and assembled into a sheet of beating cardiomyocytes. So since the primary function of heart muscle is to generate force, I first set out to characterize any contractile deficits in my cellular model of HLHS with early RV failure. So to do this, I plated the control and HLHS iPS-derived cardiomyocytes onto hydrogel micropatterns that maintain the cardiomyocytes in a 7:1 length-to-width ratio, which has been shown to be the optimal conformation for cardiomyocyte maturation and organization of the contractile apparatus. Subjectively, you may appreciate that the HLHS cardiomyocyte appears somewhat less snappy than the control. So we can then use computational video pixel motion detection to quantify the contractile properties of each individual cardiomyocyte. And we found that both the contraction force and contraction acceleration were significantly reduced in iPS-derived cardiomyocytes from all 3 HLHS patients. Now, since contractility can be affected by heart rate, I also confirmed that beat rate was not significantly different between control and HLHS. To investigate the mechanisms of the impaired contractility in HLHS, we next turned to single-cell RNA sequencing. And as we reviewed the differentially expressed genes, a couple interesting patterns emerged. So on the left, I'm showing you that genes that were overly expressed in HLHS compared to control included many, many genes that are similarly upregulated in the myocardium of patients with heart failure and cardiomyopathy. And on the right, I'm showing you that there was a marked reduction in expression of genes involved in mitochondrial function and cellular metabolism. So we then further explored the possible role of altered cellular energetics. So first, I used a mitotracker fluorescence assay to show a significant reduction in mitochondrial content in the HLHS cardiomyocytes. And this suggests that there were either fewer mitochondria or smaller mitochondria. But what we really needed to know was the functional capacity of the mitochondria in our cells. So I measured oxygen consumption rates in the presence of various small molecules that affect components of the mitochondrial electron transport chain. And what I found is that the HLHS cardiomyocytes had significantly reduced basal respiration, which includes the respiration directly related to ATP production, as well as an impaired ability to increase respiration under states of increased energy demand, as shown by the reduction in maximal respiration. So lastly, we explored the effects of altering the metabolic substrate on our cells. We treated the HLHS cardiomyocytes with our standard culture media, which is high in glucose, in comparison to a high fatty acid media. And we found a significant increase in contractility in the cells that were fed with more fatty acids. And remarkably, the magnitude of this increase was similar to what we see when we treat the cells with a bona fide inotrope, istaroxime, which increases contractility by altering calcium dynamics. And in my most recent experiments, I've now shown that mitochondrial respiration is also dramatically improved when the cells are exposed to high fatty acid versus high glucose media. So in summary, what I've shown you today is that iPS-derived cardiomyocytes from patients with hypoplastic left heart syndrome and early RV failure have an intrinsic impairment in contractility compared to controls. And this impairment is associated with reduced mitochondrial gene expression, content, and function. I also showed that metabolic substrate switching can improve contractility and mitochondrial function in the HLHS iPS-derived cardiomyocytes. And this suggests to me that impaired metabolism may represent a currently untapped therapeutic target, that could be harnessed to prevent abnormal development that leads to HLHS, as well as treat heart failure in these patients. So with that, I'd like to thank Dr. Sean Wu and everyone in his lab who's contributed, as well as our many collaborators here at Stanford and elsewhere. I'd also like to thank the leadership of the Department of Pediatrics and the Division of Pediatric Cardiology, as well as my sources of funding. And with that, I would be happy to take any questions.
Thank you, Sharon. That was an excellent presentation, really elegant work. I'm looking to see if there's a question here. It looks like there is. Is the mitochondrial defect sufficient to explain the no flow, no grow of heart failure?
Yeah, that's a really thoughtful question. Thank you for that. Congenital heart disease in general, we believe to be a polygenic and multifactorial disease. So my current hypothesis is that a valvular obstruction, so say aortic stenosis, in combination with an intrinsic myocardial deficiency, including mitochondrial abnormalities, are the two key components that would lead to HLHS. So it's those two things in conjunction that are necessary to result in such a severe phenotype.
Good answer, and setting the stage for a whole career of investigation. Lots of questions to address, which is what the fun is behind research. So congratulations again. We'll have to move on at this point in time, but thank you again for an excellent presentation. Our next speaker is Mark Wilkes. He is a postdoctoral researcher in hematology oncology. He's received MCHRI funding from the Reich Institute for Translational Medicine and the Postdoctoral Support Award, and also a SPARC Award. We also fund SPARC in part. His presentation is entitled Metformin upregulates microRNA 26A to improve erythropoiesis in preclinical models of Diamond-Blackfan anemia through suppression of Nemo-like kinase. Mark, take it away.
Hey, thank you very much. I'd like to thank the organisers for the opportunity to present our work here today. Basically, we're talking about how we're trying to inhibit this kinase, this Nemo-like kinase, in the red blood cell progenitors of Diamond-Blackfan anemia patients. So DBA is a rare disease. We only get about 25 to 30 new cases each year in the US. There's hundreds of driving mutations across at least 28 different genes. A few of these patients do have facioskeletal defects, some mild cytopenias, but the anemia is the major clinical manifestation, and this usually presents within the first year of life. So the standard of care currently is blood transfusions and steroids, both of which have many complications, issues. And then about 10 to 15% of our patients will go on to get stem cell transplants and all of the risks associated with that. So we definitely need new therapies for these patients. Now, many people in the audience probably are aware of the hematopoietic stem cell tree. So the pluripotent hematopoietic stem cell can differentiate and give rise to all the lineages of the blood. Now, in DBA, irrespective of which mutation it is, we always get this blockage that sort of occurs very early here in the earliest committed erythroid progenitors. So one of our program goals has been to try and identify a pathogenic signaling event in these progenitors that's common to all DBA patients, irrespective of their driving mutation. Now, this is summarizing a lot of work that we've done here, but basically what we found is that this kinase, Nemo-like kinase, is hyperactivated in every DBA model that we've looked at. And so just to walk you through here, we have 3 different DBA patient samples with different mutations, and we get NLK hyperactivation. Here's 2 different mouse models with different genetic backgrounds. Again, NLK is hyperactivated. And in our in vitro model, where we take healthy hematopoietic progenitors and then use lentivirus to knock down DBA genes, in each case, whichever gene we've knocked down, we always see this robust NLK activation. Perhaps more importantly, if we inhibit NLK, and in this experiment we've used siRNA, in healthy progenitors, healthy erythroid cells, we don't get any impact. And that's because NLK is not activated in this population. But if we look in the disease cells, you can see here we get a very significant increase in red blood cell production. And this is just a control now where we re-express an NLK that's resistant to the siRNA. I showed in the previous slide that we've tested NLK. NLK is active in lots of different models, and in all the models, we do get a significant increase in red blood cell production. Now, unfortunately, there is no specific inhibitor available for NLK that's out there. So we've sort of gone after this problem in a number of different ways. Firstly, as was mentioned, we've sort of teamed up over the last 18 months or so with Spark, and we've been trying to see if we could repurpose some currently approved or advanced drugs to see if we could get them that would inhibit NLK and work in our DBA models. And I'm pleased to announce that we just recently applied for a provisional patent on a compound on that front. Secondly, because NLK is a member of the MAP kinase family, we believe that by slightly modifying some of the chemical structures of known MAP kinase inhibitors, we may be able to design more specific and potent drugs. And thirdly, NLK dimerizes when it gets activated. So as opposed to going after the ATP binding sites, like most drugs, we thought another strategy might be to sort of design drugs that'll inhibit the dimerization. And we've been going after that in collaboration with SLAC. But there is a problem with this sort of standard, or a potential problem with this sort of classical drug approach, and that's that because NLK is so highly conserved with other family members, particularly in the kinase domain, we may have a challenge finding a nice specific drug. So we're really excited about this alternative approach, which takes advantage of a unique aspect of the NLK transcript. So many of you will remember that there's sort of the main protein coding region of the mRNA, but either end of that, there are untranslated regions. And if we look at the 3' untranslated region, there are sites that have these microRNA binding sequences. So what happens is when these microRNAs bind to these sequences, it targets these transcripts for degradation. And it seems that NLK is particularly susceptible to this type of regulation. So this is just a schematic now of the NLK 3' UTR, and there are tons and tons of potential microRNA binding sequences. Some of these have been characterized. This one's particularly important, MER181. It's one of the reasons that NLK is only activated in erythroid progenitors. Other groups have shown that these other ones are critical in regulating NLK expression. So we asked, are there approved drugs or supplements out there that may be able to suppress NLK expression by inducing some of these microRNAs? Again, a lot of work being sort of summarized here. We found that the diabetes drug, metformin, induces MER26, and the active component of ginseng actually induces MER208, and both of these can reduce NLK expression. So when we take this now and do it in our in vitro DBA model, you can see indeed, if we look at NLK expression, so this is a Western blot showing the level of NLK, that we reduce NLK expression when we treat with these drugs. Importantly, we don't really interfere with the expansion of non-erythroid progenitors. Again, DBA is not activated in healthy cells, and we don't see any impact of erythropoiesis or red blood cell production. But in the disease state, we either more than double and more than triple a red blood cell production in these models. So obviously now we want to move this forward and go into in vivo models. We do have a couple of different approaches. The first is standard oral therapy, but we're a little concerned that we may not get a high enough concentration of microRNAs in the bone marrow where we need to get it. So we're very excited with the potential collaboration with the Czechowicz lab, where we would like to conjugate our microRNAs with aptamers, either one microRNA or perhaps a cocktail, and that'll allow us to target these progenitors specifically in the bone marrow at a high concentration. So now just to summarize all of that, DBA is rare and we definitely need new therapies. There are hundreds of driving mutations, so we certainly need a single therapeutic target. We believe NLK serves as a strong candidate for that. However, there's no specific inhibitors out there that actually target NLK activity at the moment, although we're addressing that in a couple of different avenues. However, metformin, ginseng, and possibly other compounds that could induce the microRNAs may sort of serve as an alternative to reduce NLK expression and thereby help out this DBA patient population. With that, I need to thank my mentor, Kathy Sakamoto, everyone in the Sakamoto lab, all of our collaborators, especially our funding agencies. A big thank you to MCHRI for funding my postdoctoral fellowship. And with that, I'd be happy to take any questions or comments.
Mark, thank you very much for another excellent presentation. I'm always amazed at how much work people can squeeze into 7 minutes in terms of describing it.
Yeah, it's a challenge.
And I like your very clever approach. I think this is really innovative and important. Let me just see if there's anything that's come formally through as a question. Here's one. Let me just look here. Any other side effects of losing microRNA 26 in the body, or I guess I don't mean it's losing, it looks like it's mRNA 26 in the body. Any side effects of altering that system that you're aware of?
I don't know about high concentrations. No one's really looked at that, but I do know that both metformin and ginseng are really well tolerated drugs. Metformin treats millions of people across the world. So at least the concentration those drugs induce it doesn't seem to have major.
What about off targets? Are there things that affect?
Again, metformin's pretty well tolerated. We haven't really looked at this, mainly pediatric population. There's a chance that it might sort of impact erythropoiesis through, it also impacts AMPK and some other kinases. So there may be some off targets for sure, which is why we're excited about, our collaboration with the Czechowicz lab, where we might be able to sort of just deliver these microRNAs straight to our progenitor cells.
I think that targeting approach is a really good idea.
Really nice.
For kinds of compounds, because there probably are off targets.
You're probably right.
Yeah, really exciting work, and look forward to seeing your applications in the future as it goes on. When you even have more work, we'll have you get into 7 minutes again, see if you can.
It's 7 minutes is hard.
Okay, well, I think that ends our session. Again, congratulations to all of our speakers for really outstanding presentations. I think we can really be proud of our early career investigators and trainees. There's a good future ahead of us. I'd like to remind everybody that the session 3 of the posters is available, and I think the link will be put up for you so you can see that. Again, I'm really enjoying the symposium, and there's still more to come. Thank you very much.